All right, active transport. Now, active transport we've already defined as transport that uses a direct source of energy, okay, usually in the form of ATP. Okay, so the goal of active transport is to move substances against the concentration gradient. Okay, so when we talked about passive transport, one of the characteristics of passive transport, basically the reason passive transport, is that we move from an area of high concentration and it diffuses out into an area of low concentration. Okay, like the pine scented candle we talked about, or basically salt dissolved in water. We go from a high concentration and we move out to an area of low concentration to where everything will be equalized out. Okay, active transport is the opposite. Okay, so just imagine if we took that salt and we put it back into the cube form or got it back into the crystallized form. You can do that. You just have to boil the water off. You have to evaporate the water off, which takes energy to do. Okay, that's what active transport is. Active transport is taking things from a low concentration and condensing them or moving them to a place where it will be a high concentration gradient. Okay, and the things that typically do this in biological system, they're called pumps. Pumps, and they require energy. And the form of energy is ATP, GTP, or some kind of triphosphate. ATP is the most common. So how we're going to illustrate this is I'm going to use one of the most common pumps that we have in the human body, and it's called the sodium potassium pump. So the sodium potassium pump. Okay, it's also called the sodium potassium ATPase. Okay, so now when you see this ACE, if you've taken med term, the suffix ASE, that equals enzyme. Okay, that's all this is an enzyme. Okay, and so the ATPase is an enzyme that works on ATP, and I'll explain how it works in a little bit. So the sodium potassium pump. So first of all, what we're going to do is we are going to make our plasma membrane. I'm not going to draw a double membrane. I'm going to assume that you understand that we've got a phospholipid bilayer and stuff like that. So we're going to draw out here the extracellular. The extracellular fluid is out here, and here will be our the intracellular fluid. All right. So we only want to deal with two ions when we're talking about the sodium potassium pump, and they tell you what those are: sodium and potassium. And pretty much, if you understand sodium and potassium in the human body, you're going to be pretty set for a lot of things. Now, I'm not saying that sodium and potassium are the only ones that are important. Of course not. I would never say that. But they're gonna, for a majority of the things that the body does, it uses sodium and potassium to do it. Okay, so we've already established earlier when we were talking about the intracellular fluid and when we're talking about fluid in general, we know that the sodium concentration. So I'm going to do sodium in. We'll do sodium in red. So sodium concentration outside of the cell is high. Okay, that means there's a lot of sodium just floating around in this extracellular fluid. And if you want to do it, if you just want to draw a bunch of sodiums, okay, out here, that's perfectly fine. Okay, you can do that if it helps you visualize it. Okay, inside of the cell, there is sodium. There is sodium. So don't fret over is there sodium or isn't there sodium. Yes, there is sodium. The sodium concentration inside of the cell is low. Okay, so I'm going to draw 3 on the inside just so we can. Okay, so there's more sodium on the outside than there is on the inside. Okay, now we also established when we were talking about potassium. So the potassium concentration is low on the outside of the cell. And the potassium concentration on the inside of the cell is high. Okay, so I'm just going to draw two potassiums here. Okay, and we've got a lot more potassiums inside of the cell. Okay, then on the outside. All right, so now, fine, whoop-de-doo. Okay, so where does the active transport come in to this thing? Because you remember from when we were talking about the plasma membrane, it's selectively permeable. Ions aren't going to move through it. So why do I even need to worry about this? Well, because there are these little channels inside the plasma membrane. And there are two, there's more than two channels, but there are two of note. This is called a sodium leak channel. Okay, and through the sodium leak channel, sodium can move freely. It can move freely. So what you have to do is you have to apply the principles of diffusion, and this tells us that since I have a high concentration on the outside of the cell, got a low concentration on the inside of the cell, that the tendency of sodium is to go from the outside of the cell and it will go to the inside of the cell. Okay, and that's 100% correct. That is the tendency. Okay, because it's diffusion. Now we're dealing with two. There is another channel in there, and ta-da, it's called the potassium leak channel. And all you have to do again is apply the rules of diffusion to the potassium leak channel. You're like, okay, we got a high concentration on the inside of the cell, low concentration on the outside of the cell. The tendency of the potassium will be to move from the inside to the outside of the cell. Hey yo, that's it. Done. Okay, now put on our thinking caps. What would happen if I just left these two channels alone to do what they do? Okay, and the answer that you're going to come to is that eventually sodium concentration on the outside is going to equal the sodium concentration on the inside. The potassium concentration on the inside is going to equal the potassium concentration on the outside if we just let these do what they do. If we just let them leak. We just let them leak. Okay. Now you may be like, so who cares? And that's very valid. That's a very valid statement. Okay. The reason is we need to have these gradients. We need to have these gradients for the cell. In order for the cell to work, it has to have these gradients. It has to have a high potassium on the outside. Sorry, it has to have a high sodium on the outside, and it has to have a high potassium on the inside. Okay, especially when we talk about nerves. Nerves will not be able to conduct electricity unless we have these gradients. Okay, so now the next question is, how do I maintain these gradients with these leak channels? Okay, and I'm glad you asked. That's a very good question. Okay, so we have the sodium potassium pump. We have the sodium potassium pump. Okay, so I'll draw sodium potassium pump. Or if you remember, it's the sodium potassium ATPase. Okay, so you know the word pump. What a pump is going to do is going to move things against the concentration gradient. Okay, so I'm going to move things against our concentration gradient. So on the inside, sodium is low, outside is sodium high. So I'm going to move sodium against its concentration gradient. So what the sodium potassium pump is going to do is going to take these three sodiums, okay, and it's going to pump them out of the cell. Okay, make sense? Makes sense. And then it's going to take these two potassiums and it's going to pump them into the cell. It's going to pump them into the cell. Okay, make sense? All right, so now the problem is, you'd be like, how? How is that even possible? We don't care about whys. Whys are important. We care about how. What is the missing ingredient here? So I'm moving sodium to the outside of the cell. I'm moving potassium to the inside of the cell. Most people are like, okay, cool. What's the problem? The problem is I'm moving them against their concentration gradients. Remember, Passive diffusion occurs naturally because it uses the energy of the environment. It uses the energy of the natural motion of molecules. Okay? To move sodium against the concentration gradient is an unnatural thing. I mean, it won't happen in nature by itself. The energy isn't free energy. Okay? You have to get the energy from somewhere. Remember, that's where ATP comes in. And that's where the ATPase part of this comes in. So what the cell will do is it will take ATP and it will expend it and to turn it into ADP plus inorganic phosphate. And what that does is that is the energy to drive the pump. And so this is a very important chemical reaction that actually happens. ATP gets broken down to ADP plus inorganic phosphate plus energy. Okay? And remember, energy is the ability to do work. So I am moving sodium outside of the cell and I'm moving potassium inside of the cell. I'm doing work. Okay, it's actually moving a distance, and a force is being applied to them. Force over distance. I mean, that's the definition of work, right there, right happening, going against that concentration gradient. So that's where the energy is. Okay, so if you remember the structure of ATP, why did that happen? Okay, so if we remember the structure of ATP, it's got a phosphate, it's got a phosphate, and it's got a phosphate. You remember these two bonds. That's where the energy, well, energy is in all bonds, but in ATP, those first two bonds, those are really energetic bonds because the phosphate groups, they don't want to attach to each other. You kind of have to just force them on. And what that does is they're really cramped. They want to pop off. I mean, the third phosphate will pop off if you look at it incorrectly. You look at it the wrong way. So you can harness that energy inside of that chemical bond. Okay? And so you can think of, remember the chemical bond, this little line here, that's 2 electrons. Okay, 2 high energy electrons are sitting in there. And then that's what happens. Okay, when you pop that, when you break that, the energy from those electrons is released into the system. So we're allowed to move that. Okay, so that's primary active transport. That's active transport. That's how it works. Okay, so the takeaway from this is we have to use energy in the form of ATP in order to do work, to move things against their concentration gradients. And I think it would be beneficial to you to remember this pump. Because this pump is the main driver for their cells. You know, pretty much every one of our cells has this pump to maintain this kind of gradient. And I understand that you're like, oh, am I going to need this for my job? Probably not. But for future classes, anything in biochemistry, chemistry, biology, this pump is like the foreground, or sorry, not the foreground, but in the background of everything that you're going to study, especially in the medical sciences. You have to take this pump into account, you know, for pretty much homeostasis, keeping our cells normal. So this one. So I would take some time and to memorize this pump and figure out, you know, make sure you understand its workings. Okay. And so the 3 and 2, 3 sodiums out, 2 potassiums in. We'll talk about the significance of that when we talk about the cell itself. But I think we're spending enough time on primary. So now we're going to take a look at secondary active transport. Okay. So in primary active transport, we actually used ATP as a direct energy source. So for secondary active transport, we're going to use energy, but we're going to get it from diffusion. Okay, we're going to get from diffusion. Okay, so the best example for this is this thing called the glucose sodium cotransporter. So now cotransporter, if I cotransport, that means I'm going to move 2 things across the plasma membrane. I mean, that's what a cotransporter is. I'm moving 2 things at the same time. I'm co-transporting them. Okay, so if we look at the cell, and I'm just going to explain this, I'm going to explain cotransport as we draw it out. So if we look at the sodium glucose cotransporter, or glucose sodium cotransporter, however you want to say it. So for this, we're going to do glucose sodium co-transporter. Okay. All right, so we got our plasma membrane in here. So I'm just going to say this is extra. I want you to be able to say that's the extracellular fluid. And I'm going to call this intra. This will be the intracellular fluid itself. So we already know some things about the extracellular fluid versus the intracellular fluid itself. So we know that the sodium concentration in the extracellular fluid is high. And we know that because the intracellular sodium concentration is low. And we know from that, we automatically know from that diffusion principles are going to want to apply to this. Sodium will want to go into the cell. It really does. But remember, what's the thing that prevents it from getting inside of the cell? It's the plasma membrane. The plasma membrane will be like, no, and it will bounce off. It will bounce off the plasma membrane. Sodium does not diffuse through the plasma membrane. And remember, it's because of the lipid part. The lipid part of the plasma membrane prevents ions from going through. Okay? Glucose concentration in the extracellular fluid is typically low. Okay? And we know the glucose concentration inside of the cell is typically high. Not height, high, H-I-G-H. It's typically high. Right? And the reason is because it's bad news to have high blood glucose levels, high intracellular glucose levels. You want to get it into the cells so the cells can use it. So in order to get glucose into the cell, one of the ways to get glucose into the cell is we have to use a protein. Okay, and we're going to call it and it's called the glucose sodium co-transporter protein. So just like the facilitated diffusion proteins I showed you before, basically what this protein is a protein that's so it's an integral protein. It's an integral protein, and it's got two receptor sites. It's got two receptor sites. Okay, we're going to say one receptor site. Sorry, it's not agreeing with me today. It's got one receptor site here, and it's got another receptor site right next to it. So one receptor site at the top and one receptor site right below it. Okay? And it's basically a channel. But like all of these transport things, it's closed. Okay? So now what we're going to do is we're going to say that this top one here, and it doesn't matter which is which. We're just using this example. We're going to say this is the glucose receptor. And the bottom one here, we're going to say that's the sodium receptor. Okay? And what you have to remember is that sodium and glucose in the extracellular fluid, they're just going to be bouncing around, floating around, because remember, all molecules are in motion. And what's eventually going to happen is a glucose, I'm going to call it GLU, glucose is going to get into that receptor and it's going to bind. But nothing's going to happen. Nothing's going to happen whatsoever. Then eventually what's going to happen is a sodium is going to bind to the other receptor. It's going to bind to the sodium receptor. And then when that does, when those two things happen, the protein is actually going to change shape and the gate is going to open. And then what is going to happen is that the sodium and the glucose are going to be moved into the cell. And when that receptor empties, it's going to close again. Actually, that looks like NAT. It's not supposed to be NAT. It's Na+. Na+. Okay. That's what's going to happen. So we transported two things. Now you may be asking, and justly so, dude, glucose won't move from a low to a high. Okay, what's going to happen is the glucose is going to go out. Well, two things for this. One, it's a one-way transporter. Doesn't allow things out and only allows things in. So that kills that. And two, this is an active transport mechanism. We're moving something from a low concentration to a high concentration. Now you may be like, well, where's the ATP? Very valid question. We're using a different energy source. Okay, so remember, I'm also moving sodium from a high concentration to an area of low concentration. That is a naturally passive process. But remember, anytime you have movement, there is energy. There is energy. So what I'm doing is I'm using the energy from the diffusion of sodium, okay, from the movement of sodium. I'm using that energy, that occurs for sodium naturally, to move the glucose, okay? So that's secondary, okay? So secondary transport uses the energy usually from diffusion in order to power the movement against concentration gradients. Okay, so that's all we have on that one. Okay, so now you may be like, okay, so you may accept everything that I said, and you're like, okay, now you've introduced more sodium into this system. Won't this stabilize? Won't this equilibrate the sodium and potassium? Sorry, the sodium on the inside and the outside of the cell? And I'm like, that's a very good question, and that's a very good observation. But you got to remember, what do you have here? You got the sodium potassium pump. Over here and you got several sodium potassium pumps on the cell membrane, and what they're going to do is they're going to pick up this sodium here, and they're going to pick up two other sodiums that are floating around, these two sodiums, and they're going to be kicked out. They're going to be pumped out of the cell eventually. And you're like, okay, so in order to get glucose in, you have to expend glucose, you have to expend energy to do it. And I'm like, yes, in the end. But what's going to happen to this glucose? It's going to be metabolized to ATP, and that ATP is going to help drive this. No? Isn't that amazing? I mean, every time I think about this kind of thing, my mind just explodes. That this happens at a cellular level in all of your cells. Okay, so that's secondary active transport. Okay, and I will think we will end it there. I mean, we don't have much to do left. We'll talk about endocytosis and exocytosis in the next one, and then I think we will be pretty much done with the membrane and how I get things into the cell and outside of the cell. Right? So cool.